Okay, so we have here the term resistance. So when speak about resistance, it's just the evolutionary strategy of your organism here to resist or not to be killed in the presence of the hostile environment, even in the harsh environment that will prevent them to be killed. So they are become resistant to that. So we have here different types of resistance. The first one, we have your biologic resistance. Biologic resistance just changes that result in the organism here to that would render that one to be susceptible to be resistant to your antimicrobial agent compared before or compared previously. It would mean here na previously hindi pa siya resistant, but eventually because throughout time okay, nagiging resistant siya because of so many factors. Clinical resistance is a type of resistance conferred here by testing in the clinical trials. Okay, then we have your environmental resistance. So environmental resistance, type of resistance here, that would rendered by your microorganisms when they are exposed here to the environment that try to, number one, alter here the antimicrobial agent, or another one, it would also become resistant to the environment because of the microorganism physiologic response to your drugs. So environmental mediator here could be in the form of the number one, pH. Okay, decreased pH, acidosis would decrease the activity of your erythromycin, your clindamycin, even your aminoglycoside. Whereas increased pH, alkaline pH would render your tetracycline to eventually be okay, less effective. Another one, atmosphere, your aminoglycoside works effectively in aerobic environment. So pag hindi siya aerobic, it will not be effective. Your metronidazole, on the other hand, works best in the absence of the oxygen because anaerobic environment would convert your metronidazole into the active form. So pag may oxygen, it will not be in the active form, therefore it will not be effective. Okay, another one, we have here the concentration of your cations. We have here calcium and magnesium. So increased concentration of your calcium would try to decrease the activity of your aminoglycoside against your Pseudomonas. Whereas decreased concentration of your magnesium calcium would tend to decrease the effectiveness of your tetracycline in all types of the microorganism. Then we have here the thymidine content. Normally dapat minimal or small concentration lang ni thymidine. But if your culture media, for example, would have your high concentration of your thymidine, it would render your cotrimoxazole or the combination of your sulfonamide and your sulfonamide and trimethoprim that would render that one to be ineffective. That's false resistance. Okay, then we have the term intrinsic versus your acquired resistance. Intrinsic resistance, also called here as chromosomal or natural resistance. So that's the okay, that is governed here by the general makeup or the normal natural genetic makeup of your microorganisms. So inherent na sa kanyang character na hindi talaga siya or that microorganism is resistant to your particular antibiotics. And that natural na characteristic of the resistance here may be related to the following. Number one, impermeable. So there are some microorganisms wherein the cell wall would not allow your antibiotic to gain entry to their cell wall. They are highly impermeable. As governed here by their porins or even by their efflux. Efflux mechanism here is the parang transport mechanism that would allow parang it would efflux is palabas. Okay, so it's a mechanism here that would try to prevent the entry of your different substances, including some of your antibiotics. And some bacteria possess that activity, and therefore hindi sila magiging um, susceptible to your some of your antibiotics. Like for example, again, your vancomycin, di ba? Vancomycin is eventually will not be effective in your gram-negative because hindi siya makapasok sa gram-negative cell wall. Another one, we have your biofilm. So biofilm is a film made up of the community of bacteria. So some bacteria are able to produce biofilm that would prevent here the penetration of your antibiotics, that would render them resistant to the activity of that particular antibiotic. Enzymatic inactivation, some bacteria contains as their one of their virulence factor, the beta-lactamase. So pag may beta-lactamase sila, they try to destroy the beta-lactam ring of your penicillin, and therefore, penicillin group will not be effective. Acquired resistance, on the other hand, is type of the resistance that is being acquired by your microorganisms. Mean to say, at birth, they're not really resistant to that, but but because the product of genetic recombination, so they're able to exchange their genetic materials from one resistant or one susceptible na microorganism to try to acquire the genetic makeup of the other organism na resistant for that. So that would render that previously na susceptible na organism to become resistant as well because of their genetic exchange. They can exchange their plasmid, they can exchange their transposon, or even your insertion sequence. Those are your jumping na mga chromosome, di ba? So therefore, they try to acquire, to have your acquired resistance. Okay, we have here the different types, or we have here the examples of your natural resistance or the intrinsic resistance. First one, we have here your aminoglycosides versus your anaerobes. Remember here that your aminoglycosides works best in aerobic environment. So in the absence of the oxygen, like in the case of your anaerobe, so aminoglycoside will not be effective in the anaerobes because again, in order for the aminoglycoside to be taken up, in order for that to enter the cell here, it requires an oxygen. So, since ang anaerobe natin, walang oxygen, they try to thrive in the absence of the oxygen. Therefore, aminoglycoside will not be taken up. And therefore, once it will not be taken up, it will not reach the target. And therefore, it will not also be effective in killing your anaerobes. Then we have your vancomycin versus your gram-negative. So again, your vancomycin will not be effective against gram-negative because again, because of the lack of the uptake then. Okay, the vancomycin, sobrang laki niya that would enable your gram-negative bacteria here to take up this vancomycin. And therefore, it could not take its effects. Another one, we have your metronidazole. Metronidazole works best in anaerobic environment. Okay, in the anaerobic environment or the absence of the oxygen, your metronidazole is being converted to the active form. So therefore, effective siya. But if you have aerobic environment here or aerobes, bacteria is aerobes here, so metronidazole will not be converted to the active form and therefore it will not be effective. Okay, then we have here the different mechanism of the resistance. It might be why your microorganism will not be effective or why your antimicrobial agents or antibiotic will not be is not effective against your bacteria. Number one, enzymatic degradation like your beta-lactamase, di ba? Your beta-lactamase try to degrade your beta-lactam ring. So if the bacteria contain the beta-lactamase na enzyme, your penicillin containing the beta-lactam ring will not be effective. Another one, decreased uptake or accumulation of your antimicrobial agent inside the cell membrane of your bacteria. Pag hindi siya makapasok, it will not reach its target. Okay, so it will not eventually be effective for that. Another one, altered antimicrobial target. So, ang target nila, hindi niya makilala ni antibiotics natin. Because your bacteria try to modify the target of your antimicrobial agent. Okay, then number four, circumvention of the consequences of antimicrobial actions and uncoupling of your antimicrobial agent target interactions. Hindi siya makapag-bind. Okay, and therefore, it will not be effective. Okay, we have your specific mechanism of the resistance of your different antibiotics against certain bacteria. Number one, beta-lactam. Okay, so some bacteria will not be effective in your beta-lactam antibiotics here like your penicillin, your cephalosporin, because number one, enzymatic destruction. because those bacteria contains beta-lactamase enzyme that will degrade the beta-lactam ring of your antibiotics, of your penicillin, or even your cephalosporin. Altered target. Remember here that your penicillin, beta-lactams, and your glycopeptides, diba? So they tend to kill your bacteria. Their target is the penicillin binding protein. For the case of your beta-lactam is the enzyme. Okay, so your bacteria could eventually try to alter that one. And therefore, hindi makilala ni penicillin or other beta-lactam ring na antibiotic. And therefore, they will not able to bind to their target. And therefore, it will not cause here the cytotoxic activity with your bacteria, preventing that to be killed. Another one, decrease uptake. Your bacteria do not allow the entry of your beta-lactams. Glycopeptide also belongs to your cell wall, inhibiting your cell wall synthesis. Ang target nito, ng glycopeptide natin, would be the precursor ng ating um, penicillin binding protein. That's your D-alanine glycosyl peptidyl. So again, your bacteria will not be effective for the glycopeptide by number one, altering or try to modify the target of your glycopeptide. So therefore, pag hindi makilala ni glycopeptide ang kanyang target, it will not bind and therefore it will not cause the antimicrobial activity. Okay, another one, we have the target overproduction. Sobrang dami ng peptidoglycan. So, hindi na-overwhelm si ating glycopeptide. Hindi niya na kaya. And therefore, it will not cause here the death of your bacteria. Another one, aminoglycoside. Aminoglycoside try to inhibit your 30S ribosomal RNA, inhibiting your protein synthesis. Okay, so it could have your enzyme modification sa bacteria, could modify the site of your of the target of your aminoglycoside, or it could modify here your aminoglycoside itself. Okay, another one, decrease uptake. So they will not allow the entry of your aminoglycoside. And altered target. Okay, they try to modify their 30S ribosomal RNA. So hindi siya makilala ni aminoglycoside, and therefore aminoglycoside will not take its antimicrobial activity. Okay, then we have also here your quinolones. This is your fluoroquinolones to try to inhibit here your folic acid synthesis. Basically, this antibiotic here try to kill your bacteria by inhibiting the enzyme DNA gyrase. So, some bacteria would be resistant to your quinolones because number one, decrease uptake because the bacteria try to activate the efflux pump that would prevent the entry. Okay, pinapalabas niya ang ating quinolones and therefore it will not able to reach the target. This is the enzyme na dihydropteroate na enzyme na ating enzyme na DNA gyrase. Okay, then we have also here your altered targets. Okay, the altered target here. Okay, so the target of your quinolones, we have the DNA gyrase. So bacteria try to modify that enzyme. And therefore, it will not be recognized by your quinolones and therefore cannot able to bind and inhibit the activity of that. Okay, and therefore, hindi sila mapapatay. And lastly, we have here the macrolide. Macrolide inhibit your protein synthesis by inhibiting your 50S ribosomal RNA. So this one resistant, bacteria is resistant with your macrolide because number one of the efflux system that would pump out your macrolide, preventing the entry of that in the cell membrane. And your bacteria could also alter the target, which is your 50S ribosomal RNA, and preventing here your macrolide to recognize that one, preventing it to bind. And therefore, it will inhibit here, or it will prevent the killing of your bacteria. Okay, so thank you.